Research analysis technique in the study. This research employed a variety of techniques to investigate the antimicrobial properties of aldehydes and ketones produced during volatilization of biogenic oils. Here's the detailed breakdown of the analysis technique used. Number one is the antimicrobial activity testing. Exposure chamber method. The study utilized a controlled exposure chamber to expose bacterial cultures to known concentration of net aldehydes and ketones. So the procedure is petri dishes containing nutrients agar inoculated with Salmonella typhimurium were placed in the desiccator. A specific volume of net compounds were injected into the chamber using Hamilton syringes. The plates were exposed to a vapors of varying time intervals 1, 2, 5, 10, 15, 30, 45, 75, 90, and 120 minutes. After exposure, the plates were incubated at 37 degrees Celsius for 24 hours. The number of bacterial colonies on each plate was counted to determine the antimicrobial effect. Variation. This method was adapted to test the effect of different concentration of individual aldehydes and ketones as well as mixtures of these compounds.
This section focuses on a specific study investigating the antimicrobial activity of methyl soy vapors against biofilms formed by Staphylococcus epidermidis and Klebsiella pneumoniae. The study involved growing biofilms on microscope slides in a controlled environment and exposing them to methyl soy vapors generated at a specific temperature of 650 degrees Celsius for varying durations of 15, 30, and 45 minutes. After exposure, the biofilms were scrapped from the slides and transferred to sterile broth, which was then spatted on type TK soy agar plates and incubated at 37 degrees Celsius for 24 hours. The number of colonies on the plates was then counted to assess the effectiveness of the before treatment.
Chemical analysis. Gas chromatography flame ionization detection or GCFID. This technique was used to determine the headspace concentration of the aldehydes tested in an antimicrobial activity experiment. Procedure. A known amount of neat compound was injected to a seated desiccator. Vapor sample were withdrawn from the desiccator at regular intervals using a gas-tight syringe. These vapor samples were injected into the CGFID system. The CGFD separated the components of vapor based on their boiling points on the flame ionization detector. Quantify the amount of each component present. Purpose. This analysis helped establish the relationship between the liquid phase concentration of the aldehydes and their vapor phase concentration in the desiccator, providing insight into the effective concentration for antimicrobial activity.
Statistical analysis. Colony counts. The number of bacterial colonies on the agar plates was used to assess the antimicrobial activity of the tested compounds. The data was analyzed to determine the effect of concentration, exposure time, and bacterial strain on the bacterial survival.
The data from the GCFID analysis was used to plot graphs of concentration variance time, allowing the researchers to determine the headspace equilibrium of the aldehydes, the time required for the vapor to reach saturation.
Overall, the research employed a combination of experimental and analytical techniques to truly investigate the the antimicrobial properties of aldehydes and ketones produced during the rapid volatilization of biogenic oils. These include controls exposure experiments to assess the effectiveness of different compounds and concentrations, chemical analysis to determine the vapor phase, concentration, and statistical analysis to interpret the result.